from Microbe TV. This is TWiEVO, This Week in Evolution, episode number 63, recorded on December 30th, 2020. I'm Vincent Racaniello, and you're listening to the podcast on the biology behind what makes us tick. Joining me today from Salt Lake City, Nels LD.
Hey, Vincent. Good to be with you from LD Lab Studios.
How's it going? Everything well?
Things are good, personally. I've been watching a little bit, so instead of the weather, we've been doing the coronavirus forecasting, right? And now with the vaccines online since we last spoke.
That's right.
And so I've been following, locally, what are the new cases per day versus the new vaccinations per day.
Mm-hmm.
And right now it's about a tie. So obviously that's not good for a longer sort of view on this, how long we continue to navigate through the global pandemic. But even a few days ago, it was the cases per day were more than the vaccines per day. So if those trend lines continue, but we're going to have to hit an exponential here on the vaccines to really put a dent in this thing.
I saw a good graphic from March for Science on Instagram. It said, Operation Warp Speed at a crawl. Adequately vaccinating Americans will take 10 years at the current pace.
Yeah.
But in 1947, New York City vaccinated 5 million people against smallpox in 2 weeks. How can we be so much worse at this 75 years later?
That's right. Well, and I think the jury's still out, right? So in any massive logistical sort of situation, I think not too surprising, there'll be some kinks coming out of the blocks, but this has to change quickly and drastically if we're going to get on top of this. So as a lot of things, trying to stay a little bit patient and stay tuned, but in this case in particular, really hoping that we start to bend the curve in a different direction.
We have to. We really have to.
No
I understand logistics, Nels, because, I produce 5 podcasts and I can barely do that.
That's right. It's a lot, for sure. So, and speaking of podcasts, I've heard some rumors that there's potentially some new variants of the SARS-2 coronavirus that's been catching some attention recently.
Yeah.
Again, again, some rumpled feathers even perhaps in our science communication channels here and there as we're trying to negotiate with this new information that's just sort of bombarding us by the day. But I thought, it'd be a really good opportunity for us here at the end of the year to try to take some of the new variant information that we're all processing, that a lot of people are looking at really closely, and try to put that in a little bit of an evolutionary context and see where we end up. I think like a lot of the conversations that are happening, we're not going to have definitive answers here, but maybe a slightly different viewpoint and some comments on how we kind of navigate the days and weeks ahead and how we think about that, even from a kind of a science communication perspective as colleagues across different disciplines, and how do we sort of come together on this to some degree.
I think that would be great. I'd love to hear your perspective, because we've been talking about this over on TWiV, and I'm getting a little heat for wanting, a high standard of experimental science. And, on TWiV, we've had over the years a pretty high bar for viruses becoming more or less virulent or more or less transmissible. It's high. And now all of a sudden, people are telling me that I got it all wrong. So I said, let me talk to Nels, because he understands the lingo.
Well, I'll try to do it.
I don't mean to figure it out, but what I do object to is the mainstream media saying the new transmissible variant has arrived in Colorado, right? Because I don't think we can say that yet, but let's talk about it.
Yeah, let's talk about it. Let's try to unpack a lot of this. And, for me, it kind of brings me back a little bit to the beginning, actually, 5 years ago or plus now, when we started TWiEVO. And, so actually some of the impetus for TWiEVO, our ideas about kind of being able to cross through disciplines and, even try to translate or decode some of I mean, in science, things can get so specialized that even some of the same words mean to have 2 or 3 different definitions depending on who you're talking to. You know, in any field involving genomics, which is really at the core of trying to get a handle on what's going on with SARS-CoV-2 diversity, there is a lot to parse. There's a lot of data.
Sure.
And so, and then now, you know, given we're in the middle of a global pandemic, what might be, you know, a usual course along the lines, I think exactly that you're describing, which is what's the burden of evidence here to make a claim? This is sort of up against all of the, you know, the race to get the vaccine deployed. Here we are, you know, a year plus into the pandemic, and seeing these trend lines, regardless of you know, the good old fashioned quote unquote wild type SARS-CoV-2, some of the, and we'll unpack that a little bit too. Sort of what's the reference here? You know, even we're seeing spikes there, regardless of in case numbers, regardless of the variants, which are you know, percolating up and down. And we've had conversations about this as well. Some of the other variants that have really caught the attention of genomic epidemiologists, evolutionary virologists in particular. And so I think, you know, we won't, and as you said, and I have to concede, I'm not up to date on all of the TWiVs, which are coming.
It's all right. It's probably better that you're not.
Fast and furious. And, you know, I don't want to try to recreate that. But I do want to point our listeners that direction, because I think these conversations have been in depth. They've been really good. If you aren't already listening to this, but want to go deeper into the details here, some great conversations unfolding there. So why don't we try to unpack this a little bit? And Vincent, maybe you can help guide our conversation if I start to go into space where you're like, oh yeah, we just talked about that on the last TWiV. Folks, I think should probably go that direction. And then plus, I think, aren't you like double podcasting today? Have you already recorded another TWiV, or is that still on the menu this evening, or episode 700 in the can?
We did 700 in the can. It's not released yet, but and then I'm doing this, and I actually have another one tonight. So yeah,
triple podcasting. I
mean, I'm trying to communicate because people want information, and mainstream media, in my view, is not the place to get it. So I'm doing my best, you know.
Yeah, it's really 3 podcasts in a day. That's a lot, and parsing through all of this is even more. And so it's a really great.
Well, that's the thing, Nels.
Effort.
You can't just talk. You have to prepare. You have to read things and think about it, right?
Exactly. That's right.
So we do something. You know, Amy, my lab person, right?
Of course. Yeah.
So we started a live stream on Wednesday nights on YouTube, right? Where we just go, we turn on the cameras. We're in a format like this, and then people come. And last week we had 650 people, and they ask questions, right?
Wow, that's really
They have great questions, but it's very challenging because they ask you stuff you haven't prepared for.
That's right.
And sometimes you don't know the answer, and sometimes you get it wrong. So science live streaming is challenging. Podcasting is slightly different because you have, you know, a chance to have what you're going to talk about and read whatever you need to. But live stream is different. But I like it because the questions from people are great, right?
Yeah, absolutely. Yeah. No, and actually, getting those questions out of left field sometimes, right? From someone who hasn't trained in virology or hasn't trained in those are the, Now,
here's what we're going to do. One episode next year now is we'll live stream our recording of TWiEVO.
Yeah.
And
great.
you'll see we have this panel on eCam where all the questions come in from YouTube, and we can pick which ones, you know, we answer because they're typed in.
Sweet.
You'll really like it because, you know, they're right there, right?
Yeah, that's cool. Yeah, count me in. This sounds great. And so anyway, speaking of that, you know, so we're here again, kind of in the rough drafts of science. We talked about that earlier in the year as we were considering some of the, you know, I think this was about the time when a different variant was arising and causing some concern. This was the so-called D614G.
Right.
Mutation. And I think we'll kind of return to that in a minute. But one of the points we made was that that this was science not done in a polished form, not peer reviewed, you know, data sets that were preliminary in some cases at best. In that case, I think it's a little different in this one, but, you know, folks who are sort of cross training, maybe had never really thought about coronaviruses, but sort of jumped on the pandemic. That's, I think, less the case already a year later here. And in the case we'll be talking about of the variants in the so-called B117 lineage, which is the one that's really getting a lot of attention. This is also, considered the colloquially called the UK variant as well, for where it's been coming up in numbers. And so again, this is a work in progress. By the time this episode gets out of the can, there'll probably be a new set of preliminary data to parse through as well. But and that's why I think it's actually worth it to kind of step back and to think a little bit about this in the evolutionary context. Will the things that we talk about be a little bit more sort of not sort of that kind of caught in the headlines level? But will we sort of be pointing to some concepts in evolutionary virology and evolution in general, which will probably hold up no matter what over the test of time? And so obviously the biggest, I think, kind of headline grabbing situation here is the idea that these newly reported variants could have increased transmissibility to the tune of 50 to 70%. And so first of all, and I think again on TWiV, some really good unpacking of what that might or might not mean. If that's just on its face, if that's true, that really does deserve, in my opinion, the headline. So a virus that would have that big of an increase in transmission. And again, the definition of transmission is not necessarily the simplest thing here, even to begin with. But that could be definitely changes sort of our next several months in terms of vaccine rollout, some ideas of herd immunity, et cetera. And so it's important, I think, to get a better handle on whether or not the evidence matches those claims. So even, in the last, and maybe we should say, so just, where do these numbers come from for increases in transmission? And I would point people to TWiV. So some really good in-depth discussions on some of the limitations of the genomic and PCR-based approaches. This is different than virology, sort of traditional virology, where you can, with different experimental approaches, get a better handle on some of these numbers or some of the behaviors of virus populations. And that takes time. And so just by definition, I think it'll be probably weeks or months before we start to get evidence along those lines, which will be, absolutely crucial to stepping forward here and understanding what some of these mutations, as the virus populations very naturally are mutating and diversifying, what it might mean for things like transmission. So just sort of a kind of today on December 30th, news headline sort of report. And let me know if you're already seeing additional new evidence. But so, from last week's view, and we'll unpack a little bit of that virological preliminary report, which I think is really interesting, actually, to kind of step back and look at it from maybe a slightly different vantage point. But evidence coming out of the UK, the Nerve Tag crew, some of the consulting panels, the public health folks in the UK, and putting forward some additional evidence on this. Again, the caveat where I'm definitely not formally trained in epidemiology at all, but certainly this idea of a secondary attack rate would be a key piece of epidemiologic information on whether the virus is sort of over-replicating relative to the other variants that are out there, either by chance, by founder effect. So it's just that the virus with these set of mutations ended up in an individual or a set of individuals, a population where the human behaviors, et cetera, the spread of the virus is what is leading to these very clear numbers of sort of over-replication in some local areas, mostly in the UK, or at this point, actually pretty exclusively in the UK, versus an adaptive explanation. The virus has mutated in a way that it's adapted to become more transmissible. And so the secondary attack rate, this is defined by the number of new cases among contacts of an infected person with that virus or a reference virus, also kind of colloquially called a wild type virus, divide by total number of contacts. And for the B117 lineage, that UK variant, The numbers, I think this is comparing about 1,000 cases, again, to their contacts then, and the number of them who come back with an infection with about a 15% so-called secondary attack rate, compared to about a 9% with the non-B117 lineage, or the variants that we've seen so far. The trick here, though, at least as far as my eye can tell, as someone who's tries to take a critical, kind of constructive, critical view of the data, is that these are not matched cohorts, and so this data is vulnerable to some of the same issues about, is it founder effect versus like an adaptive outcome. And in fact, I think there are other studies, not looking at B117, but looking at sort of the viruses, populations, or variants we've become familiar with in the last year, which claims that it's about a 15% secondary attack rate, which would be basically a match to this new one. The trick is here, you can't do these you don't have a matched cohort, and then trying to do comparisons across sort of different studies falls a little bit short. And so from that standpoint, I think the jury is still out on whether, just from kind of my reading of the tea leaves of the current data, the jury is still out. Certainly other colleagues, genetic epidemiologists, who are kind of coming out a little more convinced that there is indeed increased transmissions, something in that area, to the tune of 50% or so. And again, this kind of, I think, led to some of the controversy in the last week of how we're communicating the data. And so I was thinking it might be time, Vincent. You said you have 5 podcasts running, but I think you might need a 6th, which would be TWiEPI, This Week in Epidemiology. What do you think?
If I need to get someone who can do it with me, right? And I don't know a lot of epidemiologists, and if any are listening and they have expertise and they're interested, contact me. But I would love to do it, because I think we're in this for another year at least, and a lot of issues are going to be arising. So I think it makes sense. I think no one field, as you say, has all the answers. It's about cross communication among fields, right? So I take this from a virological viewpoint. They say, we have 50% more RNA-seq reads in these patients. And I say, well, okay, I want to see some infectious virus work. And they say, well, that doesn't matter. So, but I think it does matter. So I think that would be great if we could do that. But, during this epidemic, during this pandemic, the epidemiologists are pretty busy. So.
No, of course. And adding a 6th podcast, I mean, I have a little tongue in cheek on that. Although I do think the bigger idea, so I would love to hear from more epidemiologists to unpack some of this, right? And to, and I myself have a few contacts, a few colleagues who I'd say are more genomic epidemiologists, but that would be a fascinating and important conversation to have, even if it's not an entire new podcast, certainly adding that into some of the episodes. If any epidemiologists are still talking to Vincent or willing to do so.
They're all mad at me.
Correct. But maybe the dust will settle. Some more data will come out, and then we can maybe revisit.
But that's not fair of them to be mad at me, because I'm taking a critical view as a virologist, not an epidemiologist, not a genomic. What are they? Genomic epidemiologists? Is that what they're called? Phylogenomics?
Yep, there's different sort of flavors.
It's ridiculous to be mad at me. I have spent my career, and people say I don't know anything. I think it's absurd to start getting personal. But Nels, I wanted to ask you. So just in a general theme of things, so we have these viruses move through populations which are all different. South of London is different from east of London, and so forth. So it's not surprising that you have different variants emerging, because the virus is error prone. It throws off variants all the time. And 2 things can happen. A random variant can arise, which just happens to make more progeny after it infects its cell, and that could be more transmissible, right? Or there could be pressure on the population to select such a variant from the population. If transmission is poor, maybe it will select. So there can be 2 things: random emergence and selection, right? Is that correct way to look at it?
Yeah, I think so. And I would also say that they don't necessarily have to be mutually exclusive, actually, right? So you could have sort of these two processes of the appearance of a variant in a population, or the dominance of a variant in a population could be some combination of those factors, right? There's no reason that there couldn't be, for example, human behaviors in the east of London, or some example like this, that would over promote the replication that just happened to be this variant that was hanging out there, where at the same time that variant might have emerged through some process of adaptation, and then even sort of double amplified, or or there's some balancing act there. And in fact, we're going to see that, I think, in the coming weeks, because we've heard about this case in Colorado that just kind of came in the news headlines today. And also cases in, I think, at least 20 different countries of the so-called B117 lineage starting to show up. Most of not all, but most of these cases, traced initially through travel, although that doesn't appear to be the case in Colorado. And so it's almost like a whole set now of, opportunities for that variant to then explore these populations, and this, terrible uncontrolled global pandemic that's a massively negative impact on public health. But this will tell us something about, I think, these processes of the virus spreading, or the variants spreading in the populations. Yeah.
Now, it could very well be, I want virological data, and that's why people are upset, because they say and I understand that a public health response, you can't wait, right?
Yeah.
But right. I've always said, well, if we're masking and distancing and all that properly, then we shouldn't have to do anything different, even with a variant. Nevertheless, is it possible? We have the UK variant, we have a South Africa variant, maybe even Nigeria variant. Is it possible that when those variants get elsewhere, which they have already, right? You said Colorado, the UK variant is in many places. They will not have the same advantage in a different population?
That's possible. It's also possible that, so that those very founder effect, the founder effect, I think, plays out in different places at different times in different ways. And so even if we stipulate that the virus might be more transmissible, but if it lands in Colorado, in a spot where people are like, it's a rural area, there's not much density. It's very different than London or the Kent area in the UK. Let's just say, and this is kind of speaking hypothetically here, with sort of some of the details that are actually unfolding in real time. But let's say, everyone wears masks, or for some reason that entire population got vaccinated really quickly, then it's very unlikely that even if that virus is more transmissible, that it will gain hold, just because it will, it'll be a founder effect, but that works in the opposite direction, where it didn't find anything good. There wasn't a steady stream of transmission. So that can kind of cut in both directions, actually. Yeah, definitely. So I did want to step back, and we're talking all these numbers and letters, and I guess we'll unpack that a little bit. But to that, kind of, where did this all start? And I think it was about a week ago, week and a half now, on virological.org, and the first sort of set of info that we got about this and the characterization of this. We've talked about virological a little bit before. Some of the really great, again, genomic epidemiology, a lot of the genomic surveillance, and thinking about some of the evolutionary outcomes or implications are showing up on virological. It's a good place with interesting stuff. And so this was, again, posted by Andrew Rambeau, and several other folks, kind of a big collaboration. I think the Rambeau lab was involved in setting up virological.org, kind of in a way to start to communicate a lot of this rough draft science, this preliminary science. And so
do you know Andrew Nels?
I haven't met him in person. I know one of his close colleagues, Eddie Holmes, pretty well. And they sort of came up together in evolutionary virology and have been sort of leaders in the field, I would say.
So could we get Eddie on TWiEVO?
We could. I'm sure he would do it. He's hopelessly oversubscribed.
Yeah.
Again, it's exactly what you said a few minutes ago. It would be lovely to have epidemiologists on a podcast. Yeah. They've got plenty on their plate at the moment. Eddie also lives in Australia, so doing that time change is a little bit challenging as well. But at some point, I would love to have Eddie, love to have Andrew some other folks in this space on maybe once the dust settles here on the other side of this thing, because I think we'll be unpacking this one for months, for years ahead. So, but I think it's kind of worth it to actually take a minute and go, and we'll put up the link. You can also get the link on in some of the conversation on TWiV, but to kind of take a look at the two figures here, the first two figures.
Actually, I can put them up on the video here.
Oh, sweet. Yeah, that would be great. Yeah, share the screen on that.
So I'll do figure 1, right?
Yep. Figure 1. This is a tree of some of the virus diversity. This is really zoomed in. So this is just a snapshot across. There's now just thousands of variants, into the multiple thousands, but this just shows a few of them. Yeah. And you're covering yourself there, Vincent.
Yeah, you're the man.
So, but if we look at the top left there, and if you're listening on your podcast on your, and you can get to the video of this, I think it's worth it to take a peek with us, to kind of drive through this with us. This is sort of virus variants as usual, just sort of a sampling, local sampling in and around England. So the color coding here on these dots, this is like the south end of, or the southeast of England, in not just London, just around the United Kingdom. And this is kind of what you would expect to see, is just as you're sampling folks that are infected with the virus, that there's in each of the branches indicate substitutions that are being measured in the or observed in the virus genomic sequence, the RNA sequence. And so then all of a sudden you have this on the bottom there, you have this big long branch, which leads to what we're describing, or we've called the B117 lineage, or 1.1.7, 117 for short. And you can kind of see why this would grab an evolutionary virologist, epidemiologist, sort of grab your attention, right? So what's going on here? So first of all, there's the main thing that I think really catches your eye is how long that branch is, right? And so there's all kinds of substitutions. It's kind of like this just sort of surprises you that this has, it kind of jumped. Where are the intermediate branches, actually, the missing intermediate sampling? And they might be out there, actually. And that's, to me, a really interesting evolutionary question, is how do you get from some of the strains, the reference strains, I'm using the word strain a little bit too loosely here, the original strain, and how do you get from the variants that we're seeing on here? This is all of the same strain, but how do you get to the variants that have that sort of massive jump? And because it was a massive jump, that kind of like grabs your attention. And so they have that little arrow there is actually, notating what goes into that long branch. So these are all of the mutations, the 23 mutations relative to reference. And so the reference here, I think, is actually just sort of by convention is an isolate from the first patient in Washington State. And this was right at the, I think this was last January when this genome was sequenced. And so basically this is about a year's worth of work in terms of virus evolution. And all of those dots sort of above are kind of, what we were observing in the sampling up until now, but then this sort of leap with some sort of holes in the variance between. So we might return to that point, but you could see why this might grab some attention. And so
each circle is an isolate. Is that correct?
Correct. That's right. And
the size of the circle, if there's big circle, it means more isolates of that particular kind, right?
That's right, that have been sampled. And so, and you could also look at the size of that lineage and say, oh my God, again, sampling matters here. And so, and you pick the, if this was all of the samples from, all of the United Kingdom, that would really grab your attention. But this is a small sample of them. So the fact that, the lineage though, so I guess there is something to be there is a take home message here, which is you have that long branch, and then very quickly as you're sampling, you see a lot of representatives in the lineage. And so in some ways, so the virus variant also has another name. I think it's 501Y or V. It's a lot of alphabet soup here. But in some ways, I think it's almost more accurate to speak about it as a lineage because it's spitting out variants, right? So you have all of those variants within that lineage as well. And that does speak to the fact that this is over-replicating, but, kind of with a grain of salt there. So then, we can start to look at what are those changes. So as again, kind of discussed in great detail, 8 non-synonymous or amino acid changing mutations in the spike. ORF1AB, I think 4 non-synonymous mutations. ORF8, this one is really interesting, and I think under discussed,
Yes.
where the
Agreed.
the whole gene is deleted. And in fact, we've talked about that, Vincent. So remind me, there's some other stories about coronavirus evolution that involve ORF8?
So during SARS-1 in 2003, about halfway through the epidemic, of which it was only 8,000 people, a variant of SARS-CoV-1 emerged, which lacked ORF8, and it predominated for the remainder of the outbreak. And various hypotheses were put forward. It's more transmissible, it's more virulent. But in the end, Christian Drosten did some experiments to suggest that it's actually less virulent, and maybe that's why it was more transmissible. So if you're less sick, you're more likely to be out and about and transmit. Now, this ORF8 deletions have been seen before in SARS-CoV-2, starting in Singapore and then in Taiwan during the summer. They have not predominated the outbreak as did the one in SARS-1. And there have been some reports. There's one Lancet paper which suggests these have people infected with ORF8 deletion variants have a better clinical outcome. So I agree this has been ignored in the context of the UK variant, because it could very well be that if a change makes the virus less virulent, it will increase transmissibility as a side effect, right?
Yep.
And, the limited data in the UK suggests that hospitalization rates are the same, whether you're infected with a member of this lineage or the parental lineage, but I think the numbers are too small yet. So this is a very interesting change that I think needs to get more attention. And we're focusing on spike because everyone thinks spike is the whole thing, but it's not.
Yeah, and so I think in looking at those 8 or so spike mutations, that's another very obvious place where you would hypothesize that virus adaptation could happen. And so in this case, I would say, a little bit of a circumstantial case building, because these mutations, some of those mutations have been seen in other places or even independently through kind of experimental approaches. We talked with Tyler Starr from Jesse Bloom's lab a few months back on TWiEVO about a study they did where they made this deep mutational scanning of part of the spike and then pulled out mutations based on binding to ACE2. And one of those mutations shows up in here. And so it's sort of like this lightning striking twice kind of thing. Again, it's circumstantial. And then, but also, natural occurrences. So this 501, the amino acid position 501 in the spike. So that's this other lineage that independently is arising in South Africa, and we're also seeing in some of the news headlines. This lineage is called the B1.351 lineage. And so as an evolutionary biologist, I think, as you start to see independent observations of sort of the same outcome, that definitely gets your attention. So this notion of convergent evolution, it starts to be a signal that there could be something going on here. Now, at the same time, I want to, maybe pour a little bit of cold water on getting too far ahead here on this as sort of adaptive explanation. So we've talked a number of times about this. We're talking about non-synonymous amino acid changes versus non-synonymous or changes of, so it's still a mutation, the DNA or the RNA changes, the letter-based changes, but the amino acid stays the same. And as a general rule, or as an assumption, you can imagine that those synonymous changes are sort of less impacted or less impactful in terms of adaptation, as most of the evolutionary function is working out at the protein-to-protein interface level or protein level. So, and that's a point that the authors make here, is to kind of count up the non-synonymous mutations and to note that they're overrepresented. But again, a little bit of a grain of salt here. So just because of the degeneracy of the genetic code, just by chance, or not by definition, you expect about twice as many non-synonymous to synonymous changes. And then, so there's 23. So the thing that, again, that sort of long branch is representative of 23 total mutations relative to reference, relative to that Washington 1 isolate. And so of those, but because non-synonymous are sort of more likely 2 to 1, so 6 of them are synonymous, but of the 17 that are non-synonymous, 4 of those are deletions. So if we just did a strict non-synonymous versus synonymous and multiplied by 2 to account for the degeneracy of the genetic code, now you have 13 to 12, which is basically 50-50. You can add in those 4 deletions. And if you even go back to 17 to 12, think about this like from a coin flip standpoint. If you flip a coin 29 times, total mutations here, and I'm multiplying by 2 for the silent mutations to account for the genetic code, then, the chance of hitting 17 heads or 17 tails versus 12, that's going to happen just by chance quite often.
Yeah.
So I don't think there's a strong case. So I think the convergent evolution case, independently seeing some of the same non-synonymous mutations, that gives you a little bit of a foothold from a kind of the circumstantial evidence standpoint. However, I don't think there's a strong case to be made from the number of non-synonymous to synonymous changes. That's roughly kind of what you would expect by chance.
Let me ask you a question, Nels.
Yeah.
I mean, sequencing is not equal globally, obviously, right? UK does sequencing on isolates really a lot, and we do less. In some countries, none.
Yeah.
So to what extent does sampling contribute to this? I mean, probably all of these changes have arisen before during this. It's a year into this outbreak. Why now, right? What has changed? Maybe we're just doing more sampling as part of it, and they've come up before. Now, they say that the UK variant, the lineage, never had all these together, well, as far as we know, right?
No, that's right. And I want to those are those gaps in that long branch I kind of want to return to that idea. So, so you're exactly right, which is sampling is massively uneven, especially at the genomic level. And so in the UK, they've been doing a lot of genomes to their credit. In the US, we, to our detriment, we're doing very few. And so, we're flying blind. I think if this were, this pandemic is like none other in terms of our level of surveillance. And again, I think that's why an evolutionary standpoint is really worth a lot here to try to get some context. Yeah. I mean, in the past, pandemics like this have happened, certainly with other coronaviruses, some of the common colds, and probably some of the same genetic changes. We just never had this sort of level or ability to think about trying to connect the dots down to the changes. But this raises a whole new sort of a whole new set of challenges to interpret the data, which go ahead.
I would argue that we actually don't know how to interpret them yet to a certain extent.
I agree. I think we're working this out in real time.
Yeah.
And that's kind of part of what makes this conversation in some cases, I think, a little bit awkward and tricky. And honestly, one of the big outcomes here is, is our very natural human response is as we kind of see a pattern, we really want to chase after that. And that has some trade-offs to it. So, one of the details here about it's still really hard to do a full high quality genome sequence of SARS-CoV-2. And so people are already and very, logically taking shortcuts because one of those changes in the spike coding region is a deletion. It just by chance turns out that with one of the common assays used, the PCR-based assays used, that you just, instead of amplifying a band that you can measure or quantify its rise, it just drops out, these so-called spike S dropouts or different nomenclature for this. And so people are like, oh, well, this is great. At first you look at it and you're like, oh, shoot, we can't do the normal assay that we want to run, at least for that probe. There's several probes, and so there's different ways of doing this. But because it's falling out, we can use that as a proxy to say, okay, well, if you drop out, then there's a very good chance, especially in the UK, that this is it's in the B117 lineage. Yeah. And so, that's great. On the one hand, you can very quickly sort of add more cases. You can, start to get very quickly a better handle on, is this thing going to make people more sick? And right now, the early returns are favorable that it's not making people more sick. There's downsides though. So now immediately you've changed your sampling. So the types of the numbers that go into some of the comparisons have a massive bias in that you're not doing a sort of even sampling across the board. You're very quickly saying, okay, which viruses are showing up? But if you're looking specifically for this one lineage, then very naturally, all of a sudden, everything is going to look like it comes from that lineage just because you're undersampling sort of the other isolates out there, the other variants that are out there. And so some of the data just, that's getting represented to the media, to other scientists outside of epidemiology can be misleading just in the way that the sampling is being done and the data is being collected. And so it's sort of like you for very good reasons are chasing this, but you create new problems. And again, that I think it's really important for us to be having these conversations about how do we interpret the sampling that's happening, sort of going, again, stepping back and looking at the data from sort of a bigger picture.
I think part of the problem, Nels, is that a journalist who wants to write about this, they talk to 10 scientists and 8 of them say, yeah, it's more transmissible, and 2 say no. So then they conclude it's more transmissible. They try and reach a consensus, and that's not how science works, right?
Yeah, well, the scientists' opinions or intuition doing a majority of rules seems like a little bit dangerous. No. A little bit dangerous to me. I agree with that. And of course, the challenge of talking to the media is that we as scientists are generally talking to folks who haven't trained in science at all. And so we're speaking almost in a different language. And so it's almost like this game of telephone. And so I think it's not too surprising that some crazy ideas kind of pop up, even when the scientists might have been giving a measured, very cautious interpretation or trying to represent some complicated stuff in a sort of simplified way. It can get garbled, and we're seeing that in a lot of coroners currently as well.
I had a question. This goes back just a bit, and I want to ask you because it has to do with founder effect. So, there are some individuals who say they can use computational biology to say whether a variant is emerging on founder effect or not. And I remember you saying for D614, it's really dicey to do that. So what's the story? Can you use some genomic epidemiology to say, yeah, it's a founder effect, or no? Because a lot of people have said there's no founder effect here. It doesn't happen at this point, blah, blah, blah. So.
Yeah, no, that's right. So I don't, again, kind of stipulating I'm not trained in epidemiology. My sort of layperson's impression is that you want something like that secondary attack rate, that starts to get at, so you're actually, so I think as you've pointed out, what you would love is data on human to human transmission, right? Because if it's and there are, also good reasons to go into the laboratory to do studies in ferrets, or it looks like mink might be a good choice with SARS-CoV-2 or hamsters or various things. But there's always the caveats there, right? That if it can pass from hamster to hamster, does that really apply kind of that model system to humans? Sure. So our best bet then is to go to humans. And so now we're very squarely in epidemiologic space. And so things like that secondary attack rate, right? Which is for someone who's infected, how often do they infect someone else? With that variant that they have, whether it's reference or whether it's, and so I think that's the kind of data you want, and that's the data we're waiting on. And so that first return that we just got yesterday sort of put up there, which is 15% versus 9. This is looking like 50% increased transmission, but not a matched cohort. And so I think if they match the cohort, get more data, bigger data sets, more than 1,000. If that's 80,000 matched cohort, and if it's a massive over-replicator, we'll be there in a minute or two, given our human behavior around all the other variation in SARS-CoV-2 that's really moving through populations. As unfortunately, a lot of people are traveling over the holiday season. There's going to be opportunities to get numbers here to do those comparisons. But I think I would keep an eye on, again, as a sort of an armchair epidemiologist, I would keep an eye on that secondary attack rate.
Where did this secondary attack rate data come out? Do you know?
Yeah, so this is in that updated report from the Nerve Tag, the Public Health England PHE. Yep, it's in there. I've been, one of the fun things about even the last week or two for kind of watching this story kind of back in the weeds is now I've got a new set of epidemiologists I'm following on Twitter and looking at things that they're posting. So one of the, actually I'll jump to one of these right now. So again, I picked this up through Eddie Holmes, put this out on Twitter, watching this online genomic epidemiology analysis unfold. So this is a tool that a grad student in Andrew Rambo's lab, her name is Amy O'Toole, and she's in Edinburgh, Scotland, where the Rambo lab is. She has it's got a great acronym. Some of these acronyms are a little out there, like Nerve Tag. It seems like it should be in a James Bond movie to me or something like that. But hers is GRINCH, Global Report Investigating Novel Coronavirus Haplotypes. So again, you have to very much account for the uneven sampling, and in fact, the even potentially more biased sampling that's happening. But watching as more and more genomes are slowly dropped of SARS-CoV-2 in sampling with a particular eye on the rise or fall of lineages like B117 or the variant out of South Africa 1351. And so far, and she's updating it every day. I think she did one today. Today's the 30th when we're recording. And so for both of those lineages, she states the same caveat. Most locations outside the original focus have not reported sustained transmission. In many cases, have known travel links to the focal location. Increasing numbers of international cases is currently likely due to increased surveillance and vigilance. And so this is that, I think this is to me, I think this is like, it kind of puts us on that solid footing of, what is the sort of evidence you need to really conclude that transmission versus founder effects versus epidemiological sort of variables like that are at play here.
I mean,
so,
that's secondary attack rate, basically. When a variant goes to another country, you just see how well it infects people, right?
That's right. And so this is happening. This is happening at least in 20 countries. And as we heard last night, happening across the Rocky Mountains from me in Colorado. But, the thing is, Vincent, again, and you brought up this point, which is that our surveillance is so sort of Swiss cheese and weak compared to the UK that I think you could even mount an alternate hypothesis that B117 started in the US. Someone flew over to the UK. They did so much more testing that now we think it came from the UK, but it could be smoldering right here in our backyard. So that might be a little bit of an extreme hypothesis, but certainly what we do know is that for this Colorado case, that person hadn't traveled to the UK. And so that implies that there's at least some level of transmission here. So this will be really, I mean, even by the time I'm guessing that this podcast is put out there, this will already be obsolete. We'll already know more. It'll kind of push our hypothesis in one direction or another. But then now, and as pointed out in that Grinch tool, which I'm kind of taking a look at it every day now to see as it updates.
So what do you look at, Nels, specifically on Grinch?
Yeah, so if we just kind of click on there, and we'll put the link in our show notes, of course. So it goes through the two lineages. I'll just focus on the 117 since we're kind of focusing on that one, and it's a number of figures. So how many times do you see that variant showing up as sequences are being deposited?
I see.
As genomes are being deposited, what are the dates? Put onto maps to see, how is this thing, at least, and again, I think we've talked about, when we talked about nextstrain.org, which is one of the major repositories, we've talked about some of the dangers of mapping this and how you can be fooled again by sampling. But it does start to give you an overview of what's going on here. And then the sequence count per country. So this starts to get at that secondary attack rate, potentially, again, with those caveats of sampling. But currently there's 4 known cases, for example, in Australia. If we come back in a month, is it still only 4? Or is there, or is every case coming out of Australia now the B1 from the B117 lineage? Those are the kind of patterns I think that will start to really put this on a more firm footing. And this is in Norway, India, Netherlands, Denmark, kind of across the board, Sweden, the U.S. now, which is not yet even updated on here, because that Colorado case I think they're just doing the full genome now, or trying to, but they haven't deposited yet into the main database. So the U.S. isn't on here, even though we've heard that in the news reports. So anyway, but then you could start to see counts per country, rolling averages in the frequency, and that this starts to get at it. And there's even some interesting data, kind of complementary data on air traffic from the UK to different destinations. So you can start to kind of correlate it. Again, sort of building this circumstantial case depending on what the outcomes are. And I think the jury very much still out, very early days for this. But I want to jump back actually to the, to that ORF8 loss, for example, that, like as you're saying, might make the virus more transmissible. It might even be, you could imagine a hypothesis where there's some combo of mutations here where the ORF8 deletion might be a milder case, but that's somehow weirdly balanced by some spike mutation that gives the virus and we just don't know. We're in such early days here. But so that part of the biology will be really slow to come. But there's already, I think, been some really interesting hypotheses put forward, including in that virological report, about how do you make these leaps? How is it that the virus kind of sort of jumps to new mutational space? And so if we actually scroll down to figure 2, there's another representation of that B117 lineage compared to the other samples that were coming out at the same time.
Okay.
And the graph here, it's the date of collection.
I'm going to add that one here. I'm going to add that to our video here.
Yeah. Let's do that screen share. Actually, I'm going to grab some water.
Yep.
And yeah, there it is, right? So you're looking at that regression line, that lower line, which is sort of, that's the equivalent on that phylogenetic tree of the upper left corner. And there you're seeing like that variation over time. So on the y-axis, average per site genetic divergence from root. And so that's, again, that you start at the reference strain. And then ooh, that's delicious. Over time, you can see, mutations accumulate. As you've noted at many times, as many virologists have noted, viruses mutate. So, but here, look at lineage 117. All of a sudden, collected at the same time, all of these variants in the lineage kind of jump off that regression line, right? And that's that kind of surprise. I mean, that I can see why this grabbed the attention of epidemiologists, of virus evolutionists.
So does this mean that the mutation rate per site is increased?
Probably not. So that's, I mean, I think it's a hypothesis worth considering. Is this a mutator strain? And, as I think you've pointed out in the ORF1A-B, there are non-synonymous changes.
Yeah.
So the thing is that I think it's preliminary data. If that were true, that slope of that regression line, it shouldn't be in parallel to the other sampling, right? It should be more steep. And so at least from those early samples, and they're all kind of clustered together because there just hasn't been that much time, you don't see that. You see a lot of those dots, but you don't. But the slope of the line looks about the same.
Okay, got it.
So then the question becomes, how do you make that leap? How do you get off of the regression line? And so the authors of this piece put forward the notion of maybe this is emerging from an immunocompromised individual.
Yeah.
And I think it's an interesting idea. So we've talked about this actually a couple episodes ago on TWiEVO, this report from Adam Loring at Michigan, TWiEVO 61, shot and chaser of SARS-CoV-2 evolution. And they pointed out, right, there's this pretty large population of humans who are immunocompromised in one way or another, undergoing cancer treatments that necessarily have to knock back their immune system. Autoimmune disease obviously says something about how our immune system responds to challenges like the pandemic. And so evidence certainly that in some of these cases, some of these patients, that they can shed virus, they can for a very long time, into the hundreds of days. And so kind of potentially a chance, and with a lot of treatments. So things like monoclonal antibody treatment, which might select for some of the variants that come out here. So I think an interesting hypothesis, not just Adam's paper, but preprint, that paper, a couple other papers that also kind of raise that idea and do show some mutations. Although it got me thinking a little bit, Vincent. So I'm going to launch an alternate hypothesis here based on, very thin or no evidence at all.
Okay.
But it got me thinking, so maybe that's right. And I think that is worth pursuing, is what can happen as we continue to make observations out of immunocompromised folks. So the conclusion of Adam's study was that there were some mutations, but there wasn't sort of this, jump off of the curve kind of a thing happening. And I'd be curious to see if other people actually, are able to observe something like that. An alternate I think that might be worth considering is, given that coronaviruses actually have a relatively low mutation rate. This is not influenza. This is not HIV in terms of point mutations. But one thing that they do a lot of is recombination. And so imagine if a person was co-infected with different variants of coronavirus, SARS-CoV-2, that recombined. And then you could imagine, I think, putting together different sets of mutations very quickly. And so, the first obvious question is, does this happen? So I did a quick search right before our show, and it got me onto it because of that ORF8 story that you reminded me of in the Singapore cases. So that Lancet paper, looking at clinical outcomes of cases of the ORF8 deletion in SARS-CoV-2, echoing on SARS-1 being a more mild infection, and with that conclusion. So they actually, when they were genotyping the samples that they were recovering from those patients, they noted that about 8% of the cases actually also, in addition to the ORF8 deletion, which is like a 380 base pair deletion, they were also able to pull out wild type virus genomes. And so in that 8 or 10% of patients, it's a relatively small study, 130, about 10% actually had it looks like had two variants that they were infected by. So now imagine that if you're, as the virus is spreading, and people are colliding more and more, that it's possible that you could be a mixing pot for two different variants. If they undergo recombination, all of a sudden, let's say you have 3 variants in this guy, 3 mutations in this guy, 3 in this. Now you come together, you have 6. And this would be, the rest of the genome is basically identical. So the recombination event itself is scarless. You just recombine, but you sort of then mix and match the mutation. So anyway, that's my favorite pet hypothesis at the moment for how you get to that leap or that long branch to a lineage like B117.
So Nels, if you're doing short read sequencing on patient samples, you may not pick up multiple infections. You're going to get confused, right?
Exactly. And so the magic of that ORF8 deletion is it's a lot easier to genotype because there's this big 380 base pair deletion.
Yeah.
So you can easily figure out that you're co-infected. And so if it was 0, then, throw my hypothesis out the window potentially,
right.
or that would, but the fact that it's 10% has me a little bit curious that this could be a way to get at lineages like this, and something that, I'll probably start in the new year. I'll probably start badgering some of my lab folks to try to take a peek under the hood. I don't know if you could even, given the sampling that's out there, could you start to try to piece together events to recreate what happened with B117? Are there sort of local variants that if you combine them in various ways, you start to get close to that sort of strange collection of 23 mutations? I think it's worth considering. But again, I've thought about this for about 20 minutes, so I don't want to get too far over my skis on that one. So I think that's kind of where I am on this one, which is, I think, as we've discussed, a circumstantial case building, perhaps for transmission, all of the founder effect stuff. I think open questions. I'm really going to have an eye out. I'm following Amy O'Toole's tool to see if the Grinch visualization, to see what's happening with sort of both of these lineages as we get more sampling. A little bit of a, caution, is the sampling, how biased is the sampling? To me, it's an echo when we think about like the public health measures, like when currently in the US, we don't allow people to fly from the UK to the US unless they have a test, or if, in cases where you start to shut down borders between countries. I think these kind of panic responses can actually have weird side effects where all of a sudden, we saw this, I think, in early months where travel from Europe was closed down to the US.
Yeah.
And what happened? Everyone crowded into airports and got out of the country as fast as possible in an unorganized way. Everyone got stacked up at JFK for hours, all, breathing on each other. We just, as humans, we just make the craziest sort of panicked decisions sometimes. And so, unfortunately, I think we're seeing some echoes of that, but, also I think some signs of hope as we're watching the vaccines hopefully ramping up. And we'll, I'm sure, be speaking about that into the new year. That will be sort of one of the major efforts of 2021.
Nels, I want to ask you a little bit about the South Africa lineage. So an independent lineage of a variant that has a lot of changes combined, distinct from the UK. It emerged in South Africa.
Yeah. And
their hypothesis, this is a paper, a preprint just came out a couple of days ago. They say one hypothesis is that the lineage arose through intra-host evolution in one or more individuals with prolonged viral replication. And they say this hypothesis is supported by the long branch length connected the lineage to the remaining sequences, which you showed us for next tag, right? N501Y, which is present in the South African lineage, is one of several spike changes that have emerged in an immunocompromised individual in the U.S., which is the paper we discussed of Adam Loring.
Yeah.
It should be noted, however, that the observed diversity within this lineage cannot be explained by a single long-term infection in one individual because the lineage contains circulating intermediate mutants with subsets of the main mutations that characterize the lineage.
Yep.
And so if evolution with long-term infections explains this lineage, one would need to invoke transmission chain that passes through multiple individuals. So not just one immunocompromised individuals, but multiple ones. What do you think about that?
Yeah, so I think either you need, I think it's plausible. Either you need multiple individuals or you need multiple virus variants in the same individual or some combination of both, right? But I think, at least from what I'm seeing, the missing ingredient here is sort of recombination and sort of trying to recognize that happening in real time. And it's hard because these are scarless. And so the fact, if you recombine, it's like, you've got virus genome 1 and virus genome 2. You kind of walk along and then you recombine and you just go into the second half of virus, and that at that junction right around it, the sequences could be completely identical. And it's just the variants on, the so-called left and right ends of the genome where you would put them together in new ways. I think that's part of the puzzle here. When I see that jump in figure 2 on that virologicalorg posting, or when I see those long branches where sampling to date, which, hopefully is relatively unbiased, the fact that we didn't pick that up, that to me feels like a plausible explanation. Immunocompromised, perhaps long-term, either multiple individuals and or multiple viruses, multiple variants of viruses coming together in an individual who could be immunocompromised, sort of the best of both worlds for a virus, potentially. Yeah.
Interesting how, we never thought about this. And now suddenly we get the one report from the US and maybe others. And it's similar to the situation with polio vaccine, the oral polio vaccine, which replicates in people's guts. And we learned years ago that in immunocompromised people, it can replicate in the intestine for years, for decades, actually. And you have extensive evolution that you can measure and clock it so you know how many years have been in there. And that's a problem because it evolves closer to wild-type polio in terms of its neurovirulence, right? And not something we would have expected, but it's there. It's in the literature. So you would think that we would consider this, right?
Well, there's a lot to consider all in real time. And so obviously, and again, I guess maybe my final point, kind of to bring it up to date today, is a end of year media briefing that the World Health Organization just put out in the last few hours, which is basically stating SARS-CoV-2 is not necessarily the big one when it comes to pandemics. And so, I think we can think about this on a couple of different levels. So one is, let's not put all of our attention on B117. There could be a new lineage, percolating in the weeds here,
of course,
that we'll see next week. And so, I think we can learn from this example, but sort of keeping that surveillance when if we're immediately jumping onto S dropouts as a way of trying to get a sense of what's going on, that has some drawbacks. So taking that big view, and then honestly, keeping our eyes open for future pandemics. Are we going to sort of in the, what's looking like a success story, at least from the efficacy of some of the vaccines that are now online, still holding our breath on distribution. But will we be able to quickly translate those advances? So whether it's if a new strain of SARS-2 does actually emerge, not just a variant, but one that defeats the immune responses to this one. And so, one of the things I think that we should have as we're kind of crystal ball gazing a little bit is one thing we do know is that given a good vaccine distribution, we will put really strong selection on SARS-CoV-2. And so, hopefully that selection will take a couple of years, 3, 4, who knows how many years to sort of play out, where we're thinking about, do we need to do a new vaccine? But sooner or later, that's going to happen. And so, instead of so that's one of the, I guess, my take home messages for today's podcast is, are we taking that longer view? Are we balancing our sort of necessarily very short-term kind of actions and efforts, but also keeping in mind the bigger picture here for the current pandemic, but then future ones ahead?
Yeah. I would like to close by saying, if you go to the CDC and WHO websites and read their statements on these variants, they say they may be more transmissible. They do not conclude that they are more transmissible, because as we have told you in this episode, we need more data on that. And I think those are the people to listen to. And, many news outlets are concluding that it's, I saw yesterday, the more transmissible variant has arrived in Colorado, which I think is really not appropriate to conclude that. And that's what I'm trying to do in all of my communications, is to not jump the gun and not panic people, and just listen to the CDC and WHO, what they're saying. Now, they've both had issues in this pandemic, I understand, but that is a measured way to look at these variants, and don't conclude more than the data tell you.
Yeah, I think that's about where I'm sitting as well. I can totally see why this has grabbed the attention of epidemiologists, of virus evolutionary biologists. But yeah, I think we'll hopefully those kind of data sets will get advanced. I think that's happening, and we'll have. I think even by this time next week, we'll start to get a better picture.
I totally am for more data. I am just not right now. My feeling is the data we have do not prove increased transmission. A lot of people think so, but I don't agree. I think we need more. It's consistent with, but doesn't prove it.
Yep. There are alternate hypotheses out there. And then my last point actually is just that the current, like that wild, quote unquote wild type strain, all of those other variants, those are pretty bad. There's some interesting human psychology playing out here, which is we've almost priced in that we have this pandemic. I mean, my God, just today in the US, I think the report is 3,600 deaths or something like that.
Yeah.
Today, in the last 24 hours, in the time we've been podcasting, dozens of people dying of this pandemic. And yet we hardly blink an eyelash at that. And when we see something like this, it sort of catches your eye. I mean, it's the ongoing pandemic without this variant is bad enough. And so
that's right.
Why don't we and I think we've lost a little bit of that plot line somehow in this. And there's almost this human psychology of, well, what's the new thing that's coming at me? We've almost adapted to this and become numb to this terrible pandemic that's on us already. And so that'd be kind of my last point, is that, maybe the global pandemic volume is turned up to 9, and perhaps this variant takes it to 9.1 or something, 9.2, 9. We'll see. But having the volume on 9 for a global pandemic sucks. It's awful.
Yeah, it's horrible to begin with.
That's right. And so I think we need to keep, because the danger is we've got too comfortable. We're accepting of the virus as is. And there is a danger in sort of hopping onto the new variant in some sense, that, now I'm going to worry about this. I mean, if the outcome is that finally we wake back up, great. I don't care, but that current one is bad enough. And
totally
we're really
desperately
need these vaccines to come on. That's really what I'm looking at as our best hope here.
There is a morbid curiosity in making this pandemic worse, but as you say, it is already horrible, and we need to deal with that. I agree. Yeah. We need to mask, distance, etc., and vaccinate. And everything else is a distraction, in my opinion.
Yeah,
because if indeed this were more transmissible, you would have the same response that we already have.
Yeah, but it would be. I mean, if you just do the math, if those numbers hold 50 to 70%, those daily death rates do go up significantly, substantially, and so that matters. But so does where we are right now matter. And so, I mean, let's keep doing the science. Let's keep communicating the science, but let's also try to have some perspective. Let's have a little bit of context to the conversations. All right. Very good. So why don't we move to, I think we have a few emails. How are we on time though, Vincent? I know you've got another podcast.
I've got a few hours, so we could get through a couple of emails here. Yeah.
Okay.
No problem.
Let's knock off one or two, and then I'll let you go. But so the first email is from Ben. He writes, "Hi, Vincent and Nels. First, a TWiEVO paper suggestion. A recent study by Day et al. From Buzz Baum's lab regarding closed mitosis in association with nuclear envelope disassembly and the evolutionary implications of this. A fascinating and very TWiEVO suitable read." And Ben's got the link to this Nature paper. Yeah, Ben, this is really cool. This one caught my eye. And I love the combination, kind of the interdisciplinary between evolution and cell biology. It's sort of one of my favorite zones. And so, hoping that as in the next several months, as we move into 2021 and maybe do get a foothold on the pandemic, that will, and we have spent some time returning to other science. This is happening on TWiV as well, right? Thinking about there's other viruses in the sea, so to speak. And so there's also a lot more evolutionary biology out there. And that's a great example of a cool recent study. Second, just want to throw out my 2 cents on the evolutionary changes in a pathogen's mortality rate. I think there's 2 extra things that are really important to consider. Whether or not a pathogen may evolve to be less lethal is related to whether death is required for or impacts transmission. To the best of my knowledge, rabies is still uniformly fatal in most mammals, despite at least tens of thousands of years of evolution, probably much more. And there seems no trend toward lower mortality for rabies. Similarly, many parasites require their host to be eaten for their transmission, and so the argument could be made that this could impact a selective pressure towards greater lethality. It is also important to consider if death of the host necessarily means death of the pathogen. Many pathogenic bacteria and parasites can also be free-living, and so death of the host may not lead to death of the pathogen, or alternately, such a small proportion of the total number of organisms of the pathogen infect humans and die out that it has a negligible selective pressure. An example here could be Naegleria fowleri, a primarily free-living organism that rarely infects humans but is extremely lethal. These are the amoeba. I think you hear these news stories of brain infections when people are like swimming in ponds from time. Very rare, thankfully, but a really awful infectious disease. I love the discussion from the previous episode, and while I agree that evolving to lower lethality is probably a reasonable general assumption for most single-host obligate pathogens, the influence of mortality on reproduction and transmission should be considered. Trying to predict the future for emerging pathogens is something I love to think about and discuss with friends, and I can't wait to hear more discussion in future TWiEVO episodes. This is from Ben Liffner. Thank you, Ben, for that really thoughtful email to us. And so, this came up. I think I can't remember if this was in our last episode or a couple episodes ago in the email. And this idea, this is getting thrown out, of course, as well in the conversations about the ongoing global pandemic with SARS-CoV-2 as well. But this notion that sort of the longer there is a interaction between host and pathogen, that the pathogen becomes less virulent, becomes less, the mortality goes down. And we were discussing, I'm thinking the myxoma case, the pox virus case. Yeah, I think so. I agree. It's worth it to it's a complicated question. And I think actually one of the things that sometimes doesn't get talked about a lot around this question is the timeframes that we're considering. And I think this point is important because it also reflects, again, that notion of context or the bigger picture. And so talking kind of like in real time, what's happening in months and years versus what's happening in centuries or millennia, or eras or kind of deeper evolutionary time. I think that the trend lines or the patterns might be different. I don't think there's I definitely don't adhere to that's a hard and strict rule that pathogens will get less less virulent. And certainly in short intervals, I think it can go very much in both directions. But I wonder if there is something to that longer term pattern if you wait long enough, if you come back later enough, it's a kind of a cop out, because I can always say, oh, we'll come back in another million years, and then let's ask whether that virus or whatever is more virulent. But certainly, I think if you take that longer evolutionary view, maybe just an idea that sort of pattern becomes more predominant.
Yeah, I think the time scale has to be expanded. Thousands of years is maybe not enough. I just don't know. I mean, it seemed to be for the rabbits in Australia, a year seemed to be enough, right?
Oh yeah, that was fast. That was definitely fast.
But I don't know. I think.
Yeah. Yep.
I don't know what's going on with rabies, right? Yeah, I agree, but there's something different, and maybe the natural host of rabies doesn't always cause death, in bats. So it's something about being the unnatural host that may be an issue there. But I think for viruses in general, though, the host has to be living in order for the virus to reproduce. Other pathogens, different, right? But for viruses, there is always this balance between killing the host and not. So it's great. It's a great thing to think about. And in the current pandemic, we haven't seen any reduction in virulence. We haven't proven it, but maybe we will. Who knows? We have to keep looking.
Yeah. No, and I think, Ben, your points are really good ones about kind of considering other infectious microbes that have free living, parts of their replication cycle, or just even, long-term free living, kind of like soil kind of lifestyles. And then whether there's an analogy, if we're thinking about viruses, if there's an analogy to viruses switching hosts, whether that's the equivalent. So because they do need a living host at all times, but if you move to a different species, is that the equivalent in some regards to having sort of a free living and then returning to a host at some point, if there's some analogy to be drawn there? I think it's worth it. The question is great. I think, there's not going to be any hard or fast answer. It'll be different timescales and for different infectious microbes. But I think it's a question really worth plumbing on, because it feels like a fundamental evolutionary question, and one that obviously also captures some good attention. So, yeah, I agree. Keep those discussions with friends and others going, and we'll probably be in our future podcasting as well.
Let's do one more, okay?
Sounds good, Vincent.
This is from Shinichiro, who writes, "Hi, Nels and Vincent. I would love to hear a discussion on virulence and transmission in the context of evolution." I think we're kind of having an ongoing discussion about that, right?
Yeah, I hope so. Hopefully a little bit on the transmission, at least, for this last episode.
It would be very helpful to clarify that virulence, how sick the host becomes, and transmissibility, how well the pathogen spreads, are not always selected together. It makes perfect sense. Absolutely. Yeah. Absolutely. Virulence and transmissibility are often very difficult to separate, even in a well-controlled experimental setting. Does a large plaque indicate high virulence or high transmission on a plate? What does super enlarged spleen in a mouse indicate? Host death in 2 days or 10 days when both are 100% lethal, is this due to virulence or transmissibility? Virulence and transmissibility are confounding factors for SARS-CoV-2 human relationship. The virus is highly transmissible during the pre-symptomatic period. The disease is more virulent when viral counts wane. This likely means there is no direct selection for virulence. However, virulence, transmissibility, and host behavior are connected. Paul Ewald has some very interesting discussions and gives a link to a TED Talk and to a paper. I'm hoping is right in that how we respond public health-wise may actually reshape the evolutionary trajectory of the COVID-19 pandemic. Do nothing and maximize the spread, or mask, distance, and be kind to minimize the spread. I was thinking about this since the spring when this article came out in The Guardian, which is about how the virus may evolve to be more deadly because we're socially distancing, which I think is a kind of a flawed idea there, because as he says, transmission and virulence are different. Stay safe and keep up the fantastic work. Shin Enomoto, just a biologist who wore multiple hats.
Go ahead,
this is great. Yeah. This is absolutely perfect. And I would like to say something about this idea. The virus is highly transmissible during the pre-symptomatic period. So there is let me actually put up a figure from Daniel Griffin, which is good to illustrate this. So here we have time course of COVID. So you get infected, you get exposed. The TE is the exposure. Then you have an incubation period. Viral titers start to rise. And then when the viral titers peak, you have the viral symptom phase for about a week. And then the titers rapidly decline. And that is really the first phase. And most of these infections are quite mild. And the people who recover, that's it. And then some fraction of people go on to serious disease, which is not driven by virus any longer. It's driven by the inflammation of the host, right? Yeah. So I think all discussions about virulence are off, in a way, off the table because it's not virus driven. And we don't actually know in this early phase what determines, which people are going to go into the inflammatory phase. It would be nice if we knew that, but we don't. Yeah. So here, the evolution is unclear. Now, SARS-1, the peak of viral symptoms was during a serious disease phase, lung injury, and you were in a hospital, and that cut down transmission. So if you're very sick, right, you can cut down transmission because people are isolated, and that's not the case with SARS-CoV-2. So those are some other considerations, right, about this early phase versus virulence.
Totally agree. And I think that, that asymptomatic, that almost the domination of those asymptomatic times, again, might speak to this idea of covariant infection and the possibility of recombination. Because if you're not, when your virus loads are the highest, but you don't feel sick, you're probably still colliding with other people. And so I wonder if that could be sort of kind of at play here. And I just have to say, Shin is not just a biologist who wore multiple hats. He's one of the most creative, interesting guys hanging out at the University of Utah research community. I think currently in Ming Hammond's lab in the chemistry department. He was working with Colin Dale, who does incredibly cool work on insect symbionts, in that crossover from a insect symbiont going from a free-living bacteria to then a quote-unquote enslaved symbiont. So really fun, cool work, creative ideas, and a great letter. So thanks, Shin, for checking in with us.
Yeah, it's great. Love them.
All right, so then let's finish out with our picks of the week, science picks, and I'm all SARS-CoV-2 this time. So I was really captivated by this story. I think it's come out in a few outlets, but I've included a link here. My science pick is a Wired.com story on Caitlin Karoko's career. So she is from Hungary, worked on some of the key aspects of RNA modifications, which have become very important. They're in play in the Pfizer vaccine, similarly in the Moderna vaccine. So one of the massive problems that you create with the RNA-based vaccines is that when you put foreign RNA into a host, into a human, you spike a massive immune response. And so it turns out these modified RNA bases are sort of the key to hiding the mRNA so it doesn't just get attacked by the immune system. And so she's been working on this for decades. And this article kind of tells her story. First of all, that as a scientist immigrating to the US and sort of training in the US system, working on this, what has turned out to be obviously a massively important idea, but a pretty rocky journey in academics. So she landed in Philadelphia, worked at Temple University, moved to Penn on a tenure track position, didn't get tenure, ended up working for a while in Drew Weissman's group at Penn, where a lot of this key work in the specific modification to the RNA, those ideas were advanced, and then ended up rising at the biotech company BioNTech, which is one of the key players here on the Pfizer vaccine. And so I think some important lessons here as academics about how there are some blind spots, and how do we navigate careers, especially with ideas that don't feel familiar. Some of the most creative stuff, right? That's sort of the most visionary is going to feel the most unfamiliar. And sometimes that doesn't work. We would like to kind of hold up this idea of universities as being sort of beacons of, openness, open ideas. And yet I think when the rubber hits the road, sometimes things can fall short. So anyway, I thought I would just recommend this as a read.
Nice.
That also has really good timely implications for the vaccines that are rolling out right now.
It's a great story. Great story.
Yeah. So speaking of that, Vincent, Oh, go ahead.
Nels, I do want to say this: the title is just not right. How mRNA went from a backwater to a pandemic crusher. I'm sorry, mRNA was never a backwater, bub. I spent many years working on RNA, and many other people did. So its own function in the cell is important, and we knew about that, and we studied it. So it wasn't. It may have been a therapeutic backwater, but not a scientific backwater.
No. Well, and maybe to be to be generous to the headline writers, maybe how her work on mRNA was a backwater.
Well, that could be. It could be her work.
A crusher, but
not in general.
But I agree. Both to scientists and to biology in general, RNA is no joke. This is at the origins of life all the way through as a fundamental concept. But really interesting. But so speaking of that, I think your pick of the week sort of echoes a little bit on this.
Yeah, it does. Mine is also about mRNA, SARS-CoV-2 mRNA vaccine. I'm stealing this from TWiV because I think it's so cool, and not every TWiEVO listener is a TWiV listener. Yep. And so this is a blog post by Bert Hubert, and it's called "Reverse Engineering the Source Code of the mRNA Vaccine for SARS-CoV-2." And he takes a character-by-character look at what's making up this mRNA vaccine, from the cap to the non-coding region to the coding region. He puts it in computer terms, right? How, because I suppose he's a computer person, and he relates the DNA code to computer code, right? So, that's why he calls it the source code. And I love it. I think it's just very clever and a good way to look at it, and ends up with, so here's what it is. It's got a cap. It's got a 5' UTR, got a signal peptide. It's got an optimized version of the spike. And he talks about the proline substitution to make it in a prefusion conformation, 3' uncoding, and a poly A tail. So it's very cool. A lot of fun.
Yeah, and then you see those modified bases. They're the Greek symbol, the psi symbol, which is that sort of key modification to hiding the mRNA from the immune system. Yeah, a few, maybe some some detail quibbles on
for sure,
DNA printers versus in vitro transcription, et cetera. But it's a really fun way to think about it. And I think actually, I was kind of reflecting on this the last few days, is how coming up with that sequence, right? What is it, 4,000 letters or something like that, that go into the vaccine, or even, 4,284 characters, or take the 30,000 plus or minus characters that go into SARS-CoV-2's genome. Can you imagine, so if an editor at a journal or wherever, the newspaper, said, I'll give you 30,000 characters, write something that's gonna change the course of life in 2020. I don't think any of us could write something that's that sort of impactful, and yet that 30,000 character virus genome absolutely changed the world. And then hopefully, on an optimistic note, this 4,200 character writing in the code of the vaccine will also change the world. And so it's pretty remarkable that the efficiency of information in those in that small number of characters. Really interesting to think about. Yep.
Yeah, I think that's an interesting way to look at it. But of course, no human could ever do that. It's just not within our realm.
That's right. And so that's sort of, why do we get up in the morning as scientists and do what we do? Those mysteries of how do you pack information. So, I think it's just, there's so little we know. We might bellow on in our podcast for hours at a time, and yet when you really break it down, we're just absolutely not novices at biology. It's really exciting.
I think your article and this blog post show how interested people have gotten in science because of the pandemic. That's reflected in our listenership on the podcast, and I think it's good. And I hope part of it returns or stays with us, right? That there's a heightened interest in science because, as we've seen, we can do a lot of cool things, and we need more people working in the field.
Definitely.
In fact, our applications to the PhD program at Columbia have gone up quite a bit this year.
Is that right? That's great news. Agreed. I think we're hearing similar things about MD application pools, and I think that's a good sign for the years ahead.
It's very good. Yeah. All right, it's TWiEVO 63, last TWiEVO for 2020. We're putting 2020 behind us. We're going to move on and get rid of this pandemic in 2021. You can find the show notes at microbe.tv/TWiEVO. If you want to send us a question or comment, TWiEVO at microbe.tv. And if you like what we do, consider supporting us financially at microbe.tv/contribute. Nels Eldy is at cellvolution.org, on Twitter, Earlybird. Nels, this was our 5th year of TWiEVO. Thanks so much for doing it.
Yeah, thank you, Vincent. Happy New Year. And it's been a long decade of a year.
Yeah, it has been. I thought 2020 was going to be a great year in December, but I was wrong. I'm Vincent Racaniello. You can find me at virology.blog. Music on TWiEVO is by Trampled by Turtles. You've been listening to This Week in Evolution, the podcast on the biology behind what makes us tick. Thanks for joining us. We'll be back next month. Till then, stay curious.